Well, our next guest, our next speaker is Kevin Krull, who is on the faculty in the Epidemiology and Cancer Control Group and has the endowed chair in cancer survivorship at St. Jude. He has expertise in neuroscience, clinical neuropsychology, and biological psychology. And he's going to talk to us about the cerebrospinal fluid biomarkers in long-term neurocognitive and brain imaging adverse outcomes in survivors of childhood lymphoblastic leukemia who have been treated with chemotherapy. Welcome, Dr. Krull.
Let me say thank you to the V Foundation for inviting me to present my research, and I'll jump right into it. So the purpose of my research related to the V Foundation funding is really twofold. First is to examine protein profiles in cerebral spinal fluid at diagnoses and during chemotherapy for childhood acute lymphoblastic leukemia. And then second, to evaluate the associations between these protein profiles and neurocognitive and brain imaging outcomes that occur later. Protein patterns in the CSF may help us to identify mechanisms leading to brain injury in the ALL survivors. And if we can understand these mechanisms, we may be able to treat, or more importantly, prevent this brain injury from occurring. So let me start by talking about the evidence that reflects brain injury in long-term survivors of childhood ALL. This table comes from an investigation of roughly 212 children and adolescent survivors of ALL treated with contemporary chemotherapy-only protocol. At roughly 5 years following the completion of treatment, a significant proportion of the survivors demonstrate cognitive impairment, particularly in the areas of executive functions, such as flexibility, cognitive fluency, working memory, and organization and planning skills. These problems are persistent and limit future educational attainment and employment in the survivors. This cognitive impairment is associated with altered activation in frontal brain regions. Here you can see images of the frontal of the brain during active task completion. You notice in the frontal lobes, there are areas identified in green that demonstrate overactivity in survivors with cognitive impairment compared to survivors without impairment. We believe these brain regions are overactive because they're less efficient or less developed, and they're working harder to try to accomplish a given task. These frontal regions are likely working harder because they've not developed the same level of connectivity in neural networks or circuits throughout the brain. Here we can see 4 commonly identified neural networks represented by different colors. The color of the dots represents the common network, and the size of the dots represent the strength of the connection of that region in that given network. The brain image on the left is an average of roughly 100 survivors without cognitive problems. You can see clearly, you can identify all 4 networks, common networks, in the non-impaired survivors. The image on the right is an average of roughly 75 survivors with cognitive impairment. Notice the lack of a well-developed executive attention network, the light blue color, which you can't really see in the impaired individuals. This finding demonstrates a lack of development in this network, and thus the overactivity in the impaired cognitive function displayed in the previous slides. We're interested in identifying the mechanisms that underlie or interfere with the development of these neural networks so that we can eventually learn how best to facilitate healthy development of them. We began this quest by examining protein concentrations in cerebral spinal fluid of children prior to and during chemotherapy for ALL. This fluid is easily accessible during routine diagnostic testing and prior to intrathecal administration of the chemotherapy they routinely receive. We started by looking at concentrations of 4 proteins that signal the general health of different types of cells in the brain: astrocytes, neurons, and oligodendrocytes, which make up the myelin or the insulation on the axons of the neurons. We found that at diagnosis prior to chemotherapy, myelin basic protein from the oligodendrocytes was elevated in the CSF, suggesting that something related to the leukemia itself may be injuring the myelin that coats the axons that develop the network connections in the brain. After intensive chemotherapy treatment, nerve growth factor and tau protein increase in concentration, suggesting injury to the neuronal cell bodies and the axons themselves. This injury seems to occur within 2 months of starting the chemotherapy and likely results in the reduced network connectivity that we see later on. However, knowing when the injury occurs doesn't really help us identify the mechanisms underlying the injury. And we couldn't really identify or determine this mechanism from the small number of proteins we examined. To move this research forward, we were fortunate to secure the funding from the V Foundation to examine the entire proteome of the CSF. This proteome is the entire complement of proteins in the CSF. It can be comprised of up to 4,000 different proteins. Comprehensive assessment of the proteome content in the CSF at different times before and during therapy could inform us of mechanisms underlying the injury to the myelin and the neurons that we're starting to see. This is a complicated and time-intensive process that includes quality-controlled protein extraction, protein digestion, peptide desalting, tandem mass tag labeling, and ultra-deep high-performance liquid chromatography. All of this work is done in our mass spectrometry shared resource in the Comprehensive Cancer Center of St. Jude Children's Research Hospital. The process that we go through generates a spectrum that identifies protein signatures. You can see them here reflected along the horizontal axis, each one representing a different protein, and concentrations of the protein reflected along the vertical axis. The process is conducted on CSF samples collected at diagnosis prior to any treatment and after intensive chemotherapy, with comparisons within patients run in batches to control for potential fluctuations in internal references. Although we're still running these batches of samples, we do have some preliminary observations from the samples that have been processed on the first set of survivors. We've obtained good quality CSF with minimal contamination of blood. This is always a concern because the CSF has to be extracted from the spinal canal. Tandem labeling has progressed quite well, and liquid chromatography and mass spectrometry have identified nearly 2,500 peptides and 500 proteins in each batch. We're seeing the same 1,332 peptides and the same 339 proteins in all the batches that have been run from the different survivors so far. We've also been able to quantify the same 339 proteins in all the batches run to date across survivors. This will permit us to examine the associations between the presence of over 1,000 proteins and the change in concentration of over 300 different proteins over the course of therapy. This is a significant jump in our previous research of only 4 proteins. Knowledge of these proteins and the changes will be examined in association with the neurocognitive function in the brain imaging studies I presented previously. Although we're still running batches, we have already begun to see statistically significant changes in protein concentrations from diagnoses to post-intensive chemotherapy in the survivors run to date. And you can see these listed here in this table, showing significant differences in survivors. The initial pattern that looks like it's beginning to emerge is significantly increased inflammation in proteins that come from systemic inflammatory processes, as well as inflammation in central nervous system processes in the brain. With these processes, again, beginning within 2 months of diagnoses. I have to reiterate that this is still preliminary and we need to complete our assays on the remaining survivors. This is a smaller subset than what we'll initially complete. Survivors are also in the process of completing neurocognitive testing and the brain imaging testing that I demonstrated earlier. Still, this is a very promising start, and we hope that this will inform us as to how to intervene with these survivors to prevent the suppression of brain maturation that we see in the long-term outcomes. And hopefully we can facilitate and help these survivors thrive with enriched brain function, enriched educational attainment, enriched employment down the road. With that, I'd like to thank our survivors and the clinicians that treat them, as well as our funding sources, including the very important funding that we received from the V Foundation. Thank you.
Thank you so much, Dr. Krull. That's also another interesting presentation. It shows us the far-reaching effects of therapies over the years, so thank you so much.